Hello everyone, this is Professor Susan Chang. Today's topic is about association between trace elements and obesity-related comorbidities. We know that obesity prevalence has doubled since 1980 and continuously increased in most countries. This includes like America, Europe, Africa, and also Asia as well. So the obesity is significantly correlates with the food we eat. So that means increased in daily supply of calories since like 1980s. Metabolic syndrome is closely related to abdominal obesity. And we know metabolic syndrome is a clustering of disease, including like obesity, hyperglycemia, hypertension, high triglycerides, or low HDL cholesterol. So now we move to elements. We can subdivide its elements into macro elements and trace elements. That depends on the amount of elements in our body. So for example, trace elements are micronutrients that are essential to the human body, but present only in traceable amounts. However, inadequate supply or exposure, that means too little or too much, may increase the risk of obesity and metabolic syndrome. Also, please pay attention to trace element, this category, because under the trace element, we also have heavy metals. And heavy metal is defined as density that greater or equal to 4 to 5 grams per centimeter. And this heavy metal can be toxic heavy metals or non-toxic heavy metal. So in 1973, the World Health Organization divides 19 non-elements into 3 groups according to their function and nutrition significance. So that means we have the like essential trace elements like iron, chromium, copper, zinc, selenium, molybdenum, and iodine. Also, there's a category of probably essential trace elements like manganese or silicon, and also non-essential trace elements like aluminum, arsenic, bromine, cadmium, fluorine, mercury, and tin. So we know some of them are so-called toxic heavy metal. So chronic exposures to various metals, whether this is essential trace elements or toxic heavy metals, has become more common among the public, especially in the industrialized countries. And that is because industrialized countries increased in like vehicle pollution or like mining and smelting, and also like in agriculture, the use of pesticides also contains metal as well. And also like the women's like cosmetic like makeup also contains metal as well. Don't mention about the e-waste. So those environmental metals may contaminate our water, soil, environment, plants, and animals. So subsequently, this will accumulate in the food we eat. So we now know increased in metal exposure also increased health risk. And those environmental exposures may all contribute to this health risk. But above all, food consumption is identified as the main pathway of metal exposures, particularly consumption of contaminated crops, animal, or water from contaminated sources. And WHO has announced the top 10 chemicals of public health concern. And in this case, for example, like the first one is air pollution, the second is arsenic, and the fifth is cadmium, the 8 and 9 is lead and mercury. So 4 out of 10 are toxic heavy metal. So there is a recent meta-analysis investigating 22 articles involving 55,536 participants. So the meta-analysis investigate the association between toxic heavy metal like arsenic, cadmium, lead, and mercury, and the risk of metabolic syndrome. And what author found is that participants with metabolic syndrome had significantly higher levels of heavy metal exposure compared to those without metabolic syndrome. So author also did a subgroup analysis and looking at individual heavy metal exposure. So author found that arsenic exposure is significantly correlated with metabolic syndrome, and there is a borderline effect of cadmium and significant effect of lead exposure on metabolic syndrome, and so does mercury on metabolic syndrome. Also, author look at different types of the specimen, like blood mercury level or blood heavy metal levels are significantly associated with metabolic syndrome. So does urine or hair and nail samples. So we know toxic heavy metals has an adverse effect on our human health, and so this can we can get a clue from the like a human study, cell culture, or like animal study. For example, cadmium is known to increase lipogenesis related genes and decrease lipolysis, and also increase in pro-inflammatory cytokine like TNF-alpha or IL-beta, I1-beta, and IL-6, but also decrease the glucose transporter gene expression. So subsequently, if individuals has increased in cadmium exposure, then this will increase in the lipid accumulation, but decrease the insulin secretion and also the glucose tolerance. And the lead also has a very similar effect on glucose and lipid metabolism. So in this case, the lead exposure may result in an increase in adipogenesis, but decreased in insulin secretion and disruption of glucose homeostasis. And if we can also look at like arsenic is also have an effect on glucose metabolism. For example, arsenic exposure will increase in pro-inflammatory cytokines in the cell culture study, also decrease pipa gamma. So in this case, if one has increased in arsenic exposure, and increase in arsenic will increase the ROS activity and oxidative stress. And this may, the increase in oxidative stress together with increase in pro-inflammatory cytokine will disrupt glucose metabolism and decreased insulin sensitivity as well. So arsenic also has the same effect on lipid metabolism. So we know that arsenic exposure can also increase in ER stress, and in this case, it may alter pipa gamma and CEBP alpha pathway. So in this case, the adipogenesis pathway may be disrupted, depends on the concentration of arsenic exposure. So a recent review article by Russian scientist Alex Tinkov summarized very well about the adipotropic effects of heavy metal. So if you want to understand the adipotropic effects of heavy metal exposure, I recommend you to take a look of this review article. But to summarize this article, Alex Tinkov and a colleague propose that the adipotropic effects of heavy metals depending on the exposure concentration. For example, if one has a high exposure of toxic heavy metal in their body, then in this case, they will affect the CEBP's pathway. That means they will inhibit CEBP pathway. In this case, heavy metal may decrease adipogenesis, and this will lead to adipose tissue dystrophy. Adipose tissue dystrophy and ectopic lipid accumulation. That means it will lead to the increase of fatty liver or lipid accumulation in the organ. But if one has a low dose of the heavy metal exposure, in this case, this will increase the pipa gamma pathway. We know pipa gamma is important in the adipogenesis. So this will increase the adipogenesis and results in adipose tissue expansion and also adipose tissue hypertrophy. And in this case, this will contribute to the obesity. So that depends on the dosage of the heavy metal exposure, and this can either lead to the decrease in adipogenesis but increase in the fatty liver or lipid accumulation in the organ, or this may increase in the expansion of the adipose tissue, and this results in obesity. But what caught my attention is a recent literature which suggests there is also another association between essential trace elements, obesity, and metabolic syndrome. Because as a nutritionist, I teach essential trace elements in the undergraduate class. So what we teach students is that essential trace elements are essential for our human health. So we tend to talk about the goodness of the essential trace elements. But recent literature suggests the other way around. The essential trace elements may also promote obesity and the metabolic syndrome. So that is what attracted my attention. So for example, we have different types of essential trace element and also like macronutrients, macro elements like calcium. Thus, we know they may affect our glucose metabolism or lipid metabolism. But if the expression or concentration of these trace elements is altered in our body, and in this case, it may increase the oxidative stress and pro-inflammation, and also promote inflammation. And in this case, it may have the adverse effects on the glucose metabolism. For example, we know from the human study, animal study, and cell cultural studies, there is dose-dependent effects between the essential trace elements like zinc, copper, and iron, and the risk of metabolic syndrome. For example, in our lab, we focus on iron and metabolic syndrome. But if you look at the zinc, when we talk about zinc, we tend to say, oh, obese people has lower serum or plasma zinc level. So if they have a zinc deficiency, this will affect glucose metabolism and also affect insulin production. But if one has too much of the zinc in their body, so this increase in peripheral zinc or organ concentration of the zinc, this may also cause the toxicity to beta cells as well. And in this case, whether zinc deficiency or zinc overload can also have effect on glucose metabolism. This is the same as iron and copper. For example, if you have iron deficiency or iron overload, this can also increase in SREP1 and also disrupt pipa alpha. So in this case, this will iron overload will increase lipid accumulation, and also iron overload may increase the pancreatic beta cell death. So in this case, this will lead to the decrease in insulin secretion. However, there is still a conflict result about the role of essential trace elements and the metabolic syndrome. So there are several hypotheses being proposed to explain the association between essential trace elements or metals or elements as a whole and obesity or metabolic syndrome. For example, one hypothesis has been proposed is this may be due to the joint effects of multiple metal exposure, or so-called metal-metal interaction. And so, for example, there's a recent follow-up study involved 1,221 U.S. adult women. And so authors look at the association between metal exposure and body mass index within 5.2 years of follow-up. And the key finding from this follow-up study is that authors found there was no a single metal strongly correlated to body mass index. However, if they look at the joint effect of metal exposure, what they found is that the non-essential metals exposure was significantly positively correlate with body mass index in this 5.2 years of follow-up study. So in mixture analysis, a quartile increase in all non-essential metals was associated with higher body mass index. And that was the result from this follow-up study. Also, there's a follow-up study in China as well. So in this study, it involved 982 Chinese participants living in Yangtze River in China, and the author followed them up for 4.5 years. Again, author tried to compare the metal exposure and the risk of body mass index. And in this case, the obesity is defined as body mass index. And what you found from this quantile computation figure is that you found it showed that arsenic exposure has negative weights on BMI, but essential trace element like zinc and also manganese, boron has positive weights on the body mass index. But if author used the waist circumference as the indicator of obesity, then in this case, author found is the exposure of molybdenum, zinc, and lithium has a positive effect on waist circumference. By contrast, arsenic and iron exposure has negative effect on waist circumference. So overall, again, what author found, the poor exposure of all metals has a positive effect on waist circumference. Okay, so also there is a nationwide nutrition and health survey study in America which investigates the metal exposure and the risk effect of metabolic syndrome. And in this study, author found a dose-dependent effect of metal exposure on metabolic syndrome in American adults. For example, like a urinary arsenic, individual with the highest concentration of urinary arsenic or urinary mercury and blood selenium concentration, they all have the highest level of metabolic syndrome risk compared to the reference, which has the lowest urinary arsenic or urinary mercury or blood selenium levels. So you see, we tend to think selenium is a very good essential trace elements. But in this case, if individuals has the highest level of the blood selenium level, they also increase the risk of metabolic syndrome compared to the reference. So authors try to understand what does this result means. So they also look at whether this is due to multiple metal exposure. So in order to investigate the effects of multiple metal exposure and the risk of metabolic syndrome, they conduct a principal component analysis and try to understand the metal-metal association. So in this case, they found several several metal exposure. For example, arsenic in organic and has also increased with elemental mercury. So in this case, if individual have the highest exposure level of arsenic in organic, and it also has a higher chance of increased in elemental mercury. So in this case, if individual has these two toxic heavy metal level, then they increase the metabolic syndrome risk, high blood pressure risk, and also high triglyceride risk. But if individuals has the high level of selenium, they also tend to have the high level of zinc. So actually selenium is associated with increased zinc concentration in the body as well. So if individual has both high selenium and zinc concentration in their body, they also increase with the risk of metabolic syndrome and increase with risk of high triglycerides as well. Same for the Cooper pattern. So if individual has highest level of blood Cooper levels, then again they increase the risk of abdominal obesity compared to individuals with the lowest Cooper level. By contrast, if the individuals has the highest level of cadmium and lead, then they decrease the risk of metabolic syndrome, also decrease the risk of high blood pressure, high triglyceride, low HDL, and abdominal obesity as well. To be honest, I myself don't understand why the exposure of cadmium and lead decrease the risk of metabolic syndrome. Well, to be honest, author did not explain it well as well. Okay, so a recent study also report there is an association of individual metals and environmental risk score with metabolic syndrome. Again, this is a large study involving 3,748 adults. So in this case, they found if you look at the individual metal exposure and the risk of metabolic syndrome, there is only a nickel has decreased the risk of metabolic syndrome in individuals with the highest level of nickel compared to reference. And zinc, which is an essential trace element, so also increase the risk of metabolic syndrome. And in this case, only individuals with the highest level of plasma zinc, they increase the risk of metabolic syndrome compared to the reference, which is in the Q1. However, if you look at the overall metal exposure as the ERS, then this is very sensitively to correlate with the metabolic syndrome. So we also try to investigate the association between metal exposure and obesity and metabolic syndrome risk in our own cohort. So in this case, we investigate the exposure to 13 elements, and we focus on the body composition as obesity in size, and also we look at the risk of metabolic syndrome. Okay, because this is this manuscript is under revision, so briefly we involve 150 adults, and the 71% is male, and almost half of the participants has abdominal obesity, and almost 1/3 of participants has metabolic syndrome. So we break them down into non-essential metals and essential or probably essential metals. So we also compare the traditional anthropometry in size, like waist to hip ratio or body mass index, and also compare them to body composition in size, like body fat mass, subcutaneous fat, visceral fat, skeleton muscle mass, and also a ratio of body fat mass to skeleton muscle mass. So first of all, we found there was no correlation between metal exposure and traditional anthropometric index. And if we look at the non-essential metals, we only found like lithium exposure is positively correlated with body fat mass index. But if you look at essential or probably essential metals like chromium, copper, and manganese was also positively correlated with body fat mass, but inversely correlated with skeleton muscle mass. And so we also perform a quantile G computation model to look at the individual effects and also the pool effects of metal exposure. So if we look at the relationship between body fat mass and the essential metal, and you can see there is a positive effect of copper exposure on body fat mass. But there are negative effects of zinc exposure and molybdenum exposure on body fat mass. But again, there was no significant correlation between essential metals and body fat mass. However, there was a significant effect between non-essential metals and body fat mass. And this effect has a distinct result. For example, arsenic has a negative effect on body fat mass. But other non-essential metals like mercury, lithium, cadmium, and strontium has a positive effect on body fat mass. And we found no significant effect between skeletal muscle mass and essential or probably essential elements or non-essential elements. So next, we perform a linear regression to look at the association between metal and metabolic component. Again, if you look at non-essential metal exposure, there wasn't a significant association between non-essential metals and metabolic syndrome components, except lithium. So lithium was positively correlated with fasting plasma glucose. But we found a more significant correlation between copper, magnesium, and zinc with total cholesterol. Also, like copper and magnesium was significantly positively correlated with LDL. And by contrast, like molybdenum was positively correlated with HOMA IR index and also fasting plasma glucose. So finally, we run a multi-linear regression to predict the risk association between metal exposure and metabolic syndrome. What we found is that the essential or probably essential metals like copper and zinc was positively associated with risk of metabolic syndrome. And there is a borderline effect of cadmium on metabolic syndrome, but after the adjustment, it became a non-significant. And there was also a significant effect of magnesium on abdominal obesity. And also the copper on obesity is a borderline effect because after the adjustment of age, gender, and smoking, the effect of copper became a borderline significant, and so does cadmium as well. Okay, then we look at the dyslipidemia. So again, lithium was positively associated with risk of dyslipidemia, and also copper, magnesium, molybdenum, and zinc. So then next we do a kind of concentration effect. So we break them down according to a concentration. So in this case, the T1 is a reference, which indicates that individuals has the lowest level of that metal. and T3 is the highest level. So in the case of the zinc, you can see zinc exposure itself independently predicts risk of metabolic syndrome. But this risk is increased if the plasma zinc level increase. And this dose-dependent effect also observed in magnesium and also copper as well. But there was no such dose-dependent effect on non-essential metals. So to summarize these lectures, we know that evidence from cross-sectional, and prospective studies, they suggest exposure to both toxic and essential elements may contribute to the risk of obesity and metabolic syndrome. However, the risk of single metal exposure on obesity and metabolic syndrome remains inconsistently observed. And this is likely due to the effect of joint multiple metal exposure. Although trace elements are nutritionally essential, they can be toxic if consumed at sufficiently high levels for long enough periods. So to summarize this one, whether this is essential trace elements, we traditionally think they are important for our human health. But for the consumption level, we must know that too little that will triggers the deficient, and that has an adverse health effect as well. But if we consume too much of the essential trace elements, this can also cause a toxic effect to our human health as well. So finally, I would like to thank my students like Manisa and Farah, and also my collaborator in Russian and also in the Tri-Service Hospital as well. Thank you so much for your attention.